In order for a B cell to be activated, the antigen must cross-link the receptors, as this antigen is doing here. Note that two antigen binding receptors, two B cell receptors, are being bound by a single antigen. This is called cross-linking, and it allows for this B cell to actually recognize this as a well-bound antigen that it should potentially respond to. So let's blow this up. So you can see here that the two B cell receptors are bound to the same antigen. This is called cross-linking. This sends a signal to the side chains. Remember Ig alpha beta for both the B cell receptors? So there are multiple B cell receptors on the cell surface, and you get signaling through these side chains through a series of protein tyrosine kinases that basically leads to signal propagation to the nucleus. And it's basically the signal that tells the B cell, hey, you found antigen, do something. There is an alternative way for B cells to become activated, and that's actually using a complement component. So one of the fragments of the C3 molecule is C3d. C3d can actually also bind directly to pathogens, as it's doing here. Remember earlier when I was talking about CD markers, and I mentioned that CD21 is one of the markers of B cells? Well, that's because CD21 is actually the complement receptor 2. Now, you would think that that means it's specific for complement 2, but no, it's actually specific for C3d. It was the second complement receptor found, and it is specific for C3d. So when C3d lines a pathogen, as it's doing here, complement receptor 2, or CD21, on the surface of B cells can bind to it. In this case, CD21 is serving as that second signal, that cross-linking signal, to tell the B cell, hey, you found antigen, do something. At this point, our naive B cell has cross-linked antigen and has become activated. Activated T and B cells begin to massively proliferate in a process known as clonal expansion, which basically means that this clone of B cell with the exact same antigen receptor is going to expand many, many, many times over to make a lot of copies of itself that all have the exact same antigen specificity. Now, in a separate video, I talk about a concept known as somatic hypermutation. Somatic hypermutation actually occurs at this stage in the lymph node. So when these cells are replicating, specifically B cells, not T cells, when B cells replicate this quickly, mistakes can be made in the DNA coding region that makes up the antigen binding site. Now, this is the one part that it's okay to have mutations in, because it just ups the variability of antigen receptors that could potentially be positive for different antigens. And occasionally, we'll make antigen receptors through this process of somatic hypermutation that have a higher affinity for the antigen than the original mother cell had. And this goes into a process that we'll talk about in a separate video known as affinity maturation. So I have a separate video that talks about both somatic hypermutation and affinity maturation, which you can check out. But they both occur at this stage, right after the B cell has become active. So for now, let's keep it simple and say that our B cell is perfect. It's not going to make any mistakes. So all of these B cells have the exact same antigen specificity as this B cell. Now the cell needs to differentiate. So it's gotten a really great, strong activation signal. So the goal of B cells is to become a plasma cell. Plasma cells are specialized antibody secreting B cells. And the first antibody that is ever made anytime we begin an immune response is IgM. Why? Because the B cell, the naive B cell, is already making IgM. It uses IgM as its B cell receptor, so all it needs to do is undergo alternative splicing to turn that membrane-bound IgM into a secreted IgM. Remember, IgM has 5 chains that are held together by a single J chain. So you've got 5 IgM monomers held together by a single J chain. B cells can differentiate into IgM-secreting plasma cells in the absence of T cell help. They can do this just by strong activation and some cytokines available in the microenvironment. That's one of the reasons IgM is the first antibody that is made in any given immune response. This is also important because there are plenty of antigens out there that T cells won't ever see. For instance, if this is a bacterium, it's possible that it has lipopolysaccharide on its surface. Well, lipopolysaccharide isn't a peptide, which means a T cell is never going to see it because T cells only respond to peptides. So all of these antigens that T cells can't see, like lipids, carbohydrates, nucleic acids, glycolipids, sugars, all of these things are called T-independent antigens. T-independent antigens will only ever make IgM antibody responses because they will never have T cell help. And without T cell help, you cannot switch to any of the other types of antibodies. You can't make IgG, IgA, or IgE without T cell help. So if you can't be seen by a T cell, you're going to only make an IgM antibody response. T-independent actually stands for thymus independent, but T cell independent works just as well, and I find easier to remember. Another important point about T-independent antigens is that T cell help is actually really important for establishing the memory pool. So T-independent antigens, we don't typically have a memory response to them because IgM isn't really conducive to creating memory B cells, which that means you can't differentiate them later into IgM secreting plasma cells. We'll talk about this more when we talk about memory. Okay, so what if we want to differentiate into an IgA, IgG, or IgE secreting plasma cell? How exactly are we going to get that done? Well, the answer is obviously we're going to need some T cell help. The T cells and the B cells are going to need to interact in such a way that it provides a signal from the T cell to our B cell that says, hey, you know what would be really good for this particular antigen? Some IgA, because it's likely to be found at a mucosal surface. So I'm going to give you some directions for that. Or actually, we're going to need IgG for this one. It's pretty inflammatory. So how exactly do we make that work? Okay, so first let's reorient ourselves to the lymph node because all of this is happening in the lymph node. Remember that. So T cells and B cells will come in through the afferent lymphatic vessel, then through high endothelial venules. The T cells wind up here in the paracortex, which is the T cell zone, and the B cells wind up out here in the outer cortex where they form these little follicles. Now, remember, our B cell over here actually encountered antigen in the B cell zone. So it's in its follicle, it's encountered antigen, and now it wants to maybe differentiate into a different type of antibody secreting plasma cell. So it's going to start moving through the lymph node. So it'll go from the primary follicle to the secondary follicle. And then at the secondary follicle, it's going to get even closer to this paracortex area of the T cell zone. So close that it could actually theoretically interact with T cells that are lining the T cell zone. And that's exactly what's going to happen. All right, so let's blow this up a little bit. You have your B cell here that is now presenting antigen in the context of MHC2. And it's going to move closer to the paracortex to the T cell zone. It's not going to cross into the T cell zone, but it's going to kind of reach across. And within that zone, there are T cells that can come in and kind of interact with the B cells that are reaching across to see if the B cells are presenting their antigen of interest. And in this case, let's say the B cell and the T cell get lucky, and they see that this B cell is presenting an antigen that this T cell likes. Now, remember, if this is the first time this T cell has seen its antigen, the B cell is going to need to provide co-stimulation in the form of B7 and CD28. That's no problem, though. The B cell can easily do it. Now, once the T cell is activated, it will upregulate expression of CD40 ligand, which will ligate CD40 on the surface of the B cell. This is incredibly important for class switching. Once this interaction happens, the B cell will be able to express on its surface certain cytokine receptors that will work for cytokines that this T cell will produce. The combination of the cytokines that the T cell produces and the CD40 ligand interaction will lead to the correct signals this B cell needs to know whether or not it should become an IgA, IgE, or IgG secreting plasma cell. So it's the combination of both the CD40-CD40 ligand interaction and the cytokines that are produced in the microenvironment and by the T cell that has already been activated. Class switching occurs when an enzyme known as activation-induced deaminase, or AID, is turned on as a result of CD40-CD40 ligand signaling. AID is able to form breaks in the DNA, removing intervening DNA sequences between the recombined VDJ exon encoding the V domain and one of the new heavy chain genes that will lead to IgA, IgE, or IgG expression. So once AID works to kind of loop out that intervening DNA, you go from having IgM expression to IgG expression. There are some patients who lack CD40 ligand expression on their T cells. These patients suffer from a rare disorder known as Igm Syndrome. CD40 ligand is normally required for class switching and also the generation of memory B cells and generating the germinal center follicles where all of the proliferation of B cells takes place. The patients that have hyper-IgM syndrome are unable to class switch from IgM to any of these other classes. So they have a fully normal complement of mature B cells. These B cells all have B cell receptors that are functional, but they can't really form memory B cells, and they also can only make IgM because they can't class switch. So unfortunately, the result is that these patients typically develop severe recurrent respiratory tract infections, because without the ability to class switch, you don't make IgA, which is really important for guarding and protecting our mucosal areas. In particular, many of these infections can lead to severe pneumonias and can be life-threatening. So, in review, our naive B cell that has a fully mature receptor is cross-linked by an antigen. This occurs in the lymph node, typically by soluble antigens that have kind of percolated their way into the outer cortex where the B cells hang out. At that point, the cell becomes activated and it can clonally expand. If the cell does not receive any T cell help, it will create a T-independent antigen response. It will differentiate into an IgM plasma cell that is capable of secreting IgM antibody. If at this point it does get T cell help, then it can undergo class switching. These are the T-dependent antigens, so they're peptides. This can happen because the B cell has moved toward the paracortex where the T cells hang out, or additionally, there are T follicular helper cells that actually reside within the follicle of the lymph node. These were not really discussed in this video, but are discussed in your course note. During this massive amplification, this clonal expansion, especially with T-dependent antigens, you can get somatic hypermutation, which can influence the antigen binding sites of the B cells that are produced and therefore the antibodies that the plasma cells will secrete. When there's T cell help at this stage, you get your T-dependent antigen response, which will lead to plasma cells that are capable of secreting IgG, IgA, or IgE, and although I don't mention it, IgD, because it's very rarely secreted. These antibodies are the antibodies that you'll expect to see depending on the types of cytokines that are secreted from the CD4 positive T cell in the context of CD40 and CD40 ligand interaction.